Hi guys, and welcome to another lecture of human physiology. Today we'll be diving into chapter 2, which is the chemistry of the body, and looking at certain molecules and what they're used for, and also how things are moved about within the body. So how things get in and out of cells and that sort of stuff. So hopefully you've downloaded the lecture handout that was found in the module with this video. We're going to go ahead and transition onto that screen. So remember back from our last lecture, we talked about tissues, and remember tissues were made up of both living material and non-living material. Remember, the living stuff was the cells, and all 4 tissue types had cells in them, and all of them had this other stuff called extracellular matrix, right? And remember that extracellular matrix could be a lot or a little, depending on the type of cell or the type of tissue we were looking at. When we look at this extracellular matrix, okay, it's mainly made up of fluids and molecules within there. So polysaccharides and proteins. Right? Polysaccharides are just long chains of sugars. Saccharide means sugar, poly means many. Proteins are long structures of amino acids that we'll look at a little bit later within there. Right? So this extracellular matrix, as you can see here, is filled with stuff. Of that, most of it is liquid. It's that water. Remember, water makes up anywhere from 55 to 75% of you by mass. So there's a large component of water within your body. So, right, all these nutrients and waste products need to be able to move in and out of the cells, right, from this extracellular environment to inside the cell, intracellular, and vice versa. So when we look at the body, we're going to divide it into some compartments as far as mass. So I stated earlier, right, a good portion of your body is fluids, right? When we talk about, here in this picture, it shows 55% for males, 60% for females, and that ratio can go up even higher depending on how hydrated we are. And then the other 45% of you is solids, right? And then if we look at here, the fluid part, right, that's found within your body, this liquid, is both found inside of your cell and outside of your cell. So remember, intracellular fluid is this fluid that's inside of your cell, the stuff that's making up the cytoplasm, right? That makes up about 2/3 of it. That's where we find most of it, all right? But about 1/3 of it is outside of the cell in this extracellular environment. And of that fluid that's in the extracellular environment, all right, the majority of it is going to be interstitial, which means found in between the cells. The other 20% of it is going to be that liquid part of blood, the plasma, okay, that's making up the liquid part of the blood tissue. Remember, if we go back to last lecture, right, blood was connective tissue. So here we can see that composition. So the majority of your body, as once again, is fluid, is a liquid, right? And that liquid, we'll see, acts as a good solvent and a good medium to move things around in. That's why we're so predominantly fluid as opposed to solid. So now when we look at this composition of ECF and ICF, ECF stands for extracellular fluid, and ICF stands for intracellular fluid. So when we're looking at those fluids within there, that a gradient has to exist, okay, inside and outside of the cell. So what we mean by a gradient is, right, if you remember back to your science classes in high school or maybe in college, is, right, if we reached a point of equilibrium, is we don't really get any net movement. Nothing really moves. It's stable. It's kind of static at that point. But if we have some sort of gradient, right, there's going to be movement. And the bigger the gradient, the more movement we have. Prime example of looking at and kind of understanding gradients is going to be if I have a glass of pure water, right, and I drop a drop of red dye into it. So that red dye is highly concentrated in that drop when it enters the water, right, and the rest of the water has no dye in it, so there's a huge gradient there. And we don't need to do anything to it if we just let it sit there and come back, right, within an hour or so, that all of that water is going to have a red tint to it. It dispersed throughout there. It moved because of this gradient. Right? And in order for there to exist a gradient is within your body, we need to have a barrier between the two, right? Or else it would just always wind up being in equilibrium like that dye within the jar, and that wouldn't be any good because we need things to be able to move in and out. So as long as we have a barrier in between, which is the cell membrane between that ECF and ICF, it allows certain things to pass. Right? If we remember back from biology, we said that the cell membrane was a semi-permeable membrane, right? It's selectively permeable within there. So that means that it allows certain things to pass and other things not to pass through there. So here we can see the cell membrane. It's that phospholipid bilayer. And it's got lots and lots of things stuck in it, right? Some things pass all the way through it. Some things are just embedded within it, from proteins to channels to markers on there. There's lots and lots of things within your cell membrane. Right? So we're going to go ahead and look at how things move, right, across this cell membrane. So how nutrients, molecules, right, waste, product, all those things move in and out of the cell. And there's a couple of different ways that they do that. And the two major categories are passive transport, and then the other major category is active transport. When we talk about passive transport, we're talking about something that does not require external energy. So it doesn't require any ATP. So if you think of that analogy I made of dropping that red dye into the beaker of pure water, all right, we didn't have to add any energy to it. All right, I didn't have to stir it, I didn't have to add heat to it or anything like that. It moved on its own because of that gradient. So that's passive transport. Passive transport is going to use its own kinetic energy, we say, okay, which is the energy inherent within all atoms and molecules, and it's going to diffuse, right? So diffusion is passive transport. It doesn't require energy. So anytime you see diffusion in the name, no external energy is required to make this happen. So there's three types of diffusion. We have simple diffusion, right? And in simple diffusion, right, if we look at here, we have diffusion of things moving in and out. Simple diffusion means that small molecules are just going to pass through that semi-permeable cell wall. So they're small enough that they can fit in between the phospholipid molecules, right? So here we see a diatomic molecule, right? It's 2 atoms big, and it can get through there. Right, with simple diffusion, typically it's things that are non-polar, right? So they don't have a charge on them, or they're lipid soluble, right? Those can easily pass through. right? So some very small molecules. And they're moving from an area of high concentration to an area of low concentration. Diffusion is always high to low. Right? If you're confused about which one requires energy and which one doesn't, if we think of a ball, right? If I put a ball up on top of a hill and let go of it, it rolls down the hill on its own. All right, that's diffusion. I didn't have to add any extra energy, so it's going down its concentration gradient, right, down the hill. Whereas now facilitated diffusion, still, once again, we're going to go from a high concentration to a low concentration. It's not going to require any external energy, right? And we know both those things because the term diffusion is within it. It's facilitated. Whenever we talk about facilitated, means that we're helping it along. So in this case, right, remember in simple diffusion, only small things could fit through these little spaces. But if we've got a big chunky molecule like this, right, a protein or a polysaccharide or something, and it needs to get to the inside from the outside of the cell, it can't fit through these little spaces. So embedded within the cell membrane itself are these protein channels, all right? And these protein channels are going to help facilitate, so they make a bigger opening and allow these big molecules to move through. Right, so these allow big things which are typically lipid insoluble or charged ions, things like right, chloride or sodium, all those types of things have charges on them. Right, and some of them are like in this picture, they're what we call gated, which means you can see this one, it's closed on one end and opened on the other, right? And then once the right molecule fits into it, it opens up and moves it inside. Others are what we call non-gated, which just means they're like a tunnel. They're just a big, large opening, and anything that can fit through that opening can go through it, right? With gated it's kind of selective, right? It allows only certain shapes to go through. So our third type of diffusion was osmosis. And once again, we're talking about, since it's diffusion, right? No ATP is required from an external source. And it's going to move from a high concentration to a low concentration. All right, but what you have to understand with osmosis is it's the water that's moving, not the solute. So when we talk about solutes and solvents, right, here we have sugar water, right? So we've taken water, pure water. Water is the solvent, right? And sugar is the solute within there. So that solute, right, dissolves within the water and we don't see it anymore. But in osmosis, right, our barrier isn't going to let the sugar move, right? The sugar is higher concentration over here. And if the sugar could fit through this barrier, it would go over to this side, right? But in the case of osmosis, we're only going to let the water move. So which side of this jar has more water on it? The side that has more water is the one with the less sugar, right? That sugar took up space, right? Took up mass. So if we're equal on the top as far as our volume, then there's more water molecules over here than there is on this side. So once again, it's diffusion. So it's going to go from a high concentration of water to a lower concentration. So as we can see here, the water is going to move, right, on this side of the membrane. So hopefully that makes sense. Osmosis is that movement of water, right? So the movement of the solvent, not the solute. Right, and there's sometimes within these cell membranes are specific channels, what we call aquaporins. Aqua means water, all right, and these are specific protein channels. that only allow water to move back and forth. So we find these in things like your kidneys and your lungs, and your brain and salivary glands. So things where we're talking about lots of movement of water have these special tunnels within them that just allow water to move. So, right, if we're talking about osmosis, we have to understand this thing of tonicity. When we talk about tonicity, we're talking about the cell, right? Its tone, how the force within it, if you want to look at it that way. So in an isotonic solution, if we take cells and dump them into a liquid environment, which is like your body, right? We said that most of it is liquid. If the water content is the same on both sides, right, inside the cell and outside the cell, then we really don't have, right, we don't have a concentration gradient. So we say we are in equilibrium. So an isotonic solution, right, the solution and the cells are in equilibrium. So water is really not going to change much, right? It's the same concentration out here as it is inside of these red blood cells. So we're not going to get any real movement. Now, if we take those same red blood cells and put them in a hypertonic solution, so hypertonic solution meant, right, this water that's in the beaker had a whole bunch of solute in it. So let's say this was salt water, right, instead of this was pure water over here. And now we have water in the beaker with a whole bunch of salt in it. And the water that's inside the red blood cells is pure water. So there's more, right? If we look at, there's more water, right, as far as the concentration of water. There's a higher concentration of pure water inside the cell than outside in the beaker. So the water, or the water inside the cell, wants to travel outside into the beaker. When it does that, right, if we can think of the liquid part of the cell leaves, right, what's going to happen to the cell? It's going to shrink up, right? So it shrinks. So here we can see, right, these bottoms are actual pictures of the red blood cells. So nice red blood cells that kind of look like donuts. Here we have those that are shrinking. So if we put it in a hypertonic solution, the cell is actually going to shrink up, right? And our last, right, part C over here is we can do the opposite. So now we can put it in a solution in a beaker here. Let's say that this time, right, the liquid in the beaker is pure water, right? And the liquid, right, inside the cell, inside the red blood cell, there are things dissolved in it, right? So it's got different proteins, different ions within it that are dissolved in it. So it's not pure water in there. So we have more pure water outside the cell than we do inside the cell. So in this case, the water is going to go from a higher concentration in the beaker into the lower concentration inside the cell. So it's going to move into the cell, right? And this time the cell is going to swell up, right? And in that case, it may even swell up so much that it actually bursts, just like, right, if we're filling up a water balloon, right, there's a point where we can put too much water in it and it's going to rupture. That can happen, right, within your bloodstream if the concentration gradients got too far out of whack, right? If we remember back to homeostasis, right, there exists this, right, this balance between these two environments. So the right balance is over here when we're in an isotonic solution, right? Hypertonic, they start to shrink up and we can get too much problems, right? We can wind up being dehydrated. And the opposite is we can put those cells in a hypotonic solution, and now the water is going to move into the cell and could actually rupture it, right? Or what we call lyse, which means to rupture the cell and destroy it. All right, remember back from our original little schematic there, besides passive diffusion or passive transport, we had active transport. So active transport requires an external energy source. So you got to use energy, right? That ATP molecule, right, is our energy molecule that our body uses. You got to use ATP in order to make this happen. And when we talk about active transport, it's just the opposite of passive. So it requires energy, and it also moves things, what we say, up the concentration gradient. So it's going from a low concentration to a high concentration, right? If we think of that analogy of that ball rolling down the hill, right, that was down the concentration gradient, that was diffusion or passive transport, no energy was required. But now if that ball is sitting at the bottom of the hill, the only way it's going to go uphill, right, up the concentration gradient, so I have to put some external force into that ball, right? It can't do it on its own, right? I need to kick it. If I kick it and add an external energy source to the ball, I can get it up the hill. Same thing here. So that energy source, once again, is ATP. Right, so going back to this, some prime examples that we'll see this is when we look at the nervous system, and we talk about potassium and sodium pumps. So they're actually pumping it against the concentration gradient, right? And the reason why the body does this is it wants to have, right, it doesn't want the cell to be in equilibrium, right? By nature, everything wants to go into equilibrium, but that wouldn't be good for a lot of systems within your body. So we have to actively keep pumping them out. So equilibrium is never achieved. Right? Secondary active transport, right, are going to now be where a primary pump feeds the concentration gradient to make a second pump work. So if we look at a secondary active, here we can see, right, sodium pump maintains the concentration of sodium, right? So here's our sodium pump, right? And that was a normal primary active transport pump. It's making that we have a whole bunch of sodium out here, but this glucose now is dependent on this first pump, right? So it's a secondary active transport. So secondary active transport, right, that second pump, right, in this case, the pump to move glucose into the cell is dependent on sodium ions. If the sodium ion doesn't come in here, then this pump doesn't work, right? Once again, it required energy because this needed energy. So those are two main ways that we get things in and out of cells, right? And that's how we mostly move most small things, most molecules, and right, diatomic and small molecules like that. The other way of moving big things are going to be vesicular transport. So here we're making vesicles. Vesicles, kind of think of them like Tupperware. So there's a right, either we're moving big things like this, like a food particle, right? Or we're moving large quantities of things, right? We want to get a whole bunch into the cell or out of the cell all at once. So that's why we would use vesicular transport. And there's a couple different types, well, 4 different types on here, all right? And the first two we're going to look at are called phagocytosis and pinocytosis. With these, all right, both of these are taking molecules into the cell. So things are going from the extracellular fluid into the intracellular fluid. And what happens is the cell membrane just kind of wraps itself around, right, that molecule. And once it wraps itself around, it's able to pinch off, right, and make this new container, right, that's got the particle in it. So that's why we call them a vesicle. It made this vesicle, think of it like Tupperware, right? It brought it inside. So phagocytosis is when it's bringing in large solid pieces. Pinocytosis is when it's bringing in a fluid type of environment, Okay, so it's not a solid, it's a fluid, right? But remember, it's not pure water. It's got things dissolved in it. So that's pinocytosis. Same process. They're both bringing things into the cell, either big things or large quantities of things. Pino is bringing fluids in. Phago is bringing food in. And then sometimes we have these two other things going on, and that's a receptor-mediated endocytosis. So here, in order to make this process happen, where it does, right, endocytosis, brings stuff into the cell, is certain proteins have to land onto these receptors. And if they match, that triggers this part of the membrane to go ahead and surround this, right, bit of area and make a vesicle and bring it into the cell. So sometimes we'll look at viruses. We'll get into the cell this way, right? They have specific little things that fool the cell into thinking this is something that I want to engulf, and that's how they get in there, because viruses are pretty big, chunky things compared to a cell, right? And that's how they sneak their way in. The opposite is exocytosis. So exocytosis just means that we're going from the inside of the cell and we're dumping it out into the extracellular environment, right? A lot of the time, exocytosis is getting rid of waste products. So the cell is getting rid of waste and it's dumping it into that extracellular fluid to be picked up by the bloodstream, right? Or it could be that it's, right, had made some sort of chemical and now it wants to dump that to get into the bloodstream. So, right, we could see exocytosis with your endocrine system where it's making hormones. So the cell is making a hormone, right, and now it's going to dump it into, right, out of the cell into the environment to go to other places within the body. All right, so that's how we move things around the body. So now what we're going to go ahead and do, right, is we are going to continue on here. And we're going to now look at our next part, which is looking at classes of chemicals. So remember we said that your body was made up of living things and non-living things, right? These chemicals are the non-living structures within there, right? Both inside the cell and outside the cell, we find these structures. So we have inorganic things. Inorganic chemicals typically lack carbon-hydrogen bonds. So prime examples would be water, right? H2O. It's got hydrogen, right? But it doesn't have a hydrogen linked to a carbon. It's got hydrogen linked to, right, oxygen, right? Or carbon dioxide, right? It's got carbon, right? But it doesn't have any hydrogen linked to it. It's got oxygen linked to it. So water, carbon dioxide, oxygen, right? Acids, bases, salts, right? Sodium chloride, potassium chloride, sodium bicarbonate, right? HCl. Those are all inorganic type things, and it doesn't matter where we get those from, right? If that structure comes from, if that water comes from you, the human, right? Or you originally got it from that Arrowhead, right, water bottle, right? H2O is H2O. There isn't human H2O and non-human H2O. And then we have organic things, right? Organic things typically have carbon-hydrogen bonds to them. Remember, carbon and hydrogen was one of our four major elements that make up the majority of your body. So when we look at things with carbon and hydrogen bonding, is they're typically the structures found within your body our big molecules, or what we call macromolecules, which we'll look at in those classes. And those macromolecules are made up of smaller individual, what we call monomers. So these classes down here, carbohydrates, lipids, proteins, nucleic acids, are what we would call polymers, right? And they're made up of smaller things called monomers, right? That's one reason why when we look at, all right, is there life on another planet? These are some of those elements we look for, carbon and hydrogen, because we know these make up living things. So when we talk about organic, right, we're not talking about like organic in the food store, right, in your grocery store. It's not something that's pesticide free, right, or non-GMO. What we mean by organic here is that it's got those carbon hydrogen bonds in it, right? And typically, right, organic things, when we're talking about here in the chemistry of organic things, come from living organisms. Right? If you think about it, where do you get your carbs from, right? You don't get carbohydrates from eating a rock, right? A non-living thing. You get carbohydrates from eating plants or animals within there. Right? So when we look at water, right? Once again, we said we are anywhere from about 55 to 75% water. So it makes up a large part of our mass, right? So some of those characteristics, right? It's the most abundant chemical, right? It's really good at being a solvent. So what we mean by solvent is things can dissolve in it, right? When we put sugar into our glass of water, right? We can mix it up and the sugar seems to be gone. It's still there, right? It hasn't turned into something else. It's just broken apart into its parts, and there's water molecules in between it, right? But if we take that glass that we dumped sugar into it and dissolved all the sugar in it, we can't see it anymore, but then we evaporate all the water out, right? The sugar is left still in the glass, right? We wind up with the sugar just like we started with, right? It didn't change. It didn't turn into anything else. So that's what a solvent does, right? And when we look at water, right, as a solvent, we say that it is hydrophilic, right? So in that hydrophilic, right, hydro means water, philic means to like. So that sugar likes water, it dissolves it, right? Or water likes the sugar and dissolves it easy, or salt, right? But now if we pour vegetable oil into that water, right, it doesn't break apart, it doesn't dissolve in it. And that's because, right, there are certain molecules that it's not a good solvent for. And those things that it's not a good solvent for, we say are hydrophobic, right? Phobia means you have a fear of it, right? So that oil, right, fears the water. It does not break apart in it. It stays all clumped together. So things that dissolve in water are hydrophilic. Things that don't dissolve in it are hydrophobic. And it takes part in chemical reactions. So when we look at chemical reactions, right, excuse me, there's a couple different types. We can talk about a dehydration synthesis and a hydrolysis reaction. So hydrolysis means we're taking water in, right? We can see this bottom example here where we're taking water, right? We have two things and we're breaking them apart. It required water to do that, right? A dehydration means that we're removing the water from it, right? So typically we're taking two things, we're removing water, and when we link those two together, water comes off, right, is our molecule. So if we get dehydrated, right, water is leaving our body. So in a dehydration synthesis, right, where we're making molecules from smaller things, right, small things making big things, right? That's a dehydration synthesis. Sometimes when we're breaking things apart, right? So dissolving that salt, right? The sodium chloride into its parts, into its monomers, right? We needed water to do that. The other nice thing about water is it likes to hold on to heat very well. It's a good conductor of heat. So because of that, as it absorbs heat, it holds on to it. Okay, and it can hold lots and lots and lots of heat. That's why it's very good, right? We sweat, right? It's water that's leaving our body when we sweat to cool us down, because in each one of those little beads of water is a lot and lot of heat that's stored in it. Okay, it takes a whole, it can hold a whole bunch before it releases it. So it's very good at regulating your temperature. All right, and as we said before, things dissolve in it, so it's very good at digestion, right, and elimination. So things we can, and right, we, as we stated before, within your blood, right, the majority, right, of that liquid part within your blood is water. So it's good at circulating, moving things around. So it's a really good medium. That's why we have so much of it within our body, because of all of these properties or characteristics of it. Right. Next, we're going to look at acids and bases. So remember acids and bases, and we throw salts in here, and you'll see why in just a minute. But if we go back to chemistry class, right, acids, when they dissolve, okay, so when we dissolve an acid, it's going to give up hydrogen ions. All right, so that H plus is a hydrogen ion. Ion just means that the element has a charge to it, so either a plus or a minus. So here, H was hydrogen, right? Whereas an acid, or I mean a base, I'm sorry, when we dissolve it in water, is going to give up OHs, right? It's going to break down into OHs. So if we think of, right, if we remember back to chemistry, when we talk about acids and bases, remember we use the pH scale, right? And we said that 7 was neutral in the pH scale, and that was water. And this kind of makes sense. When we talk about something being neutral, means that it has even amounts of hydrogen ions and OH minus ions, which is called a hydroxide. So it has equal numbers. So for every hydrogen ion, it's got one hydroxide ion. If we put these two together, what do we get? H2O, right? Water. So that's why water is neutral. It means that it has even parts of a base and an acid in it, if you want to think of it on a pH scale. So now all of a sudden, if I have, right, more H's, if I have more H's than I do OH's, then it's an acid. If I have more, right, hydroxides, OH's, than I do H's within the solution, then it's a base, right? Or we could say it's alkaline is another way of saying base. So if I take these acids and bases and put them together, I typically get a salt, all right, plus water. So an example, HCl, right, is an acid. Sodium hydroxide is a base, right? So hydrochloric acid plus sodium hydroxide is going to give me sodium chloride, right, which is salt, and water. Right, so now we, right, continue on with that pH scale, that pH concept. Remember that pH scale is just talking about that relationship of hydrogens and hydroxide, right? Hydrogen ions and hydroxide ions. If they're equal, right, we're going to have water, which is neutral, right? If we have more hydrogen ions than we do hydroxides, then it's an acid. If we have more OHs, right, then it's going to be a base or alkaline. So remember, the scale goes from 0 to 14. 7 is in the middle, that's why it's neutral. Right, so the pH is less than 7, it means we have more H's. Right, so if I have two liquids and one is a pH of 6 and one is a pH of 1, one is a lot stronger of an acid than the one that's a pH of 6. All right, so the smaller the number means the more hydrogen ions we have in there. All right, it's what we call an inverse log in math. So it's inverse, which means that the smaller the number, the greater the concentration, and it's a log, right? If we remember from math class, log is a power of 10. So every whole number jump, okay, is a power of 10. So if I move from a pH of 6 to a pH of 5, it's not that that acid that has a pH of 5 is 1 time stronger than the acid that has a pH of 6, because it's a one number jump. We got to remember that every one of those whole numbers represents a 10 times fold. So that solution that's a pH of 5 is actually 10 times stronger of an acid than that pH, right, of 6. And if I went from a pH of 6 to a pH of 4, right, that's 10 to the 10th, right, 10 to the second power, right? So now that solution with a pH of 4 is actually, right, 100 times stronger than that one of 6. Not just 2 times stronger, but 100 times, right? 10 to the second power. And if we go the other way, right, if it's greater than 7, right, it means we have more OHs, more hydroxides, ions. And we say that's a base, or another way of describing a base is we say it's alkaline. And once again, as we move farther away from 7, the stronger a base it is. So a solution that has a pH of 8 and another solution that has a pH of 10, right? That solution that has a pH of 10 is a lot more alkaline, or is a lot stronger base than the one of 8. All right, in that case, it would be 1,000 times stronger, right? Because we're going 10 to the third. Right, a couple of things that I want you to know, that I want you to remember within there. Okay, there's lots of things on here. Just two that I quickly want you to commit to memory, and that is your blood. So the pH of blood, right, is 7.35 to 7.45. So that's the range, right, the homeostasis range of the pH of your blood, right? So right in the middle would be 7.4. So on average, the pH of your blood is 7.4. But if your blood was, right, like water, if it had a pH of 7, that would be outside of this range. And remember, it'd be outside of homeostasis. And that would be a problem. You're going, well, water is not a problem. It's neutral. But if your blood was pure water, right, it would be a problem. So it's a little bit basic, right, within there. And the same thing, if it got too basic, right, if it had too much stuff in it, too many electrolytes, too many other ions and stuff that was making it, right, more basic, then that would be a problem as well. So blood, I want you to commit to memory, this figure, all right. The other one, that's really the only one that I want you to commit as far as number-wise. The other ones should make sense to you, and that would be your digestive juices in your stomach, right? You don't really need to understand the value, but what I want you to see is that it's a really strong acid, right? It's in the 1.2 to 3.0 range, right? Remember, 0 was the strongest acid I could get, so it's a very strong acid within there. All right, and the other thing that I want you to notice is urine. So if we look at the pH of urine, right, it's got a really wide range. It can be anywhere from, right? 4.6 to 8.0. So sometimes your urine is acidic, right? And sometimes it's very basic. And a lot of this has to do with how much water is in it. So the more dehydrated you are, right, and you go to the bathroom and your urine is, right, a bright yellow color, means that it's, if we were to take the pH of it, it would be very acidic at this. It'd be down towards this end, right? Whereas if we, right, once again looked at urine and it was clear, almost looked like water, right, it would be very basic within there. So it'd be more towards this 8.0. So it's got a wide range. So urine can be both acidic and basic. Most of that is dependent upon how hydrated you are. So once again, 3 things I want you to know. Blood, I want you to know the actual numbers, right? Urine, I want you to know that it can be both acidic and basic, depending on how much water is in it. And digestive juices in your stomach, gastric juices, I want you to understand are extremely strong acids. All right, so organic compounds. When we talk about organic compounds, right, once again, we're talking about those things that, right, have carbon and hydrogen bonds to them, right? Typically, we say they're structurally complex, right? So a carbohydrate is a lot more chemically complex than an oxygen molecule or a water molecule or a salt molecule within there. So many, they're very big structures, right? So they don't easily, right, move in and out of cells, right? These are these things that typically we look at, that exocytosis and endocytosis, right, of moving these quantities in and out of our body, in and out of our cells, I mean, within there, or using facilitated diffusion or even sometimes active transport. Right, once again, right, our classes that we alluded to before are carbohydrates, lipids, proteins, and nucleic acids, right? And if you think of those first 3, carbs, lipids, and proteins, right, that makes up that food pyramid that we talk about, right, that we want to make sure that we're eating every day, because these make up a large part of what your body needs. So first one we're going to go back is look at carbohydrates. All right, carbohydrates within there contain carbon and hydrogen because they're an organic molecule, and they also have oxygen bound to them. When we think of carbs, carbs are our major energy source for our body, right? So we have that plate that we're eating for dinner, and it's got carbs, lipids, and proteins on it, right? It's got some carbs, it's got some proteins, it's got some fat mixed within it. Of all that food on your plate, what your body is going to use first for energy are going to be all the carbs that you consume. Right? When we talk about carbs, they are soluble in water, right? Therefore, if we use that terminology, they're hydrophilic, right? They like water. They dissolve in water. We take sugar, right, which is a simple carbohydrate, it dissolves in water, right? So carbohydrates we break down into simple and complex, right? Typically, simple carbohydrates are, oops, let me go back here. Sorry about that. Simple carbohydrates are sugars. So sugars scientifically are known as saccharides. So a monosaccharide or a disaccharide, right, are these simple sugars within there. So things like glucose, fructose, galactose, right, maltose. And if you notice all those sugars that I just named, they all ended in OSE. So typically if we see a word and it ends in OSE, we know it's a simple carbohydrate, right, or a simple sugar. And those give us quick energy, right? We can use those very quickly. We can break them down and turn those into ATP very easily. So if we need, right, energy real quickly, right, it makes sense to grab that. Whereas the other type of carbohydrate are complex carbohydrates. And these types of carbohydrates are made up of, right, a whole bunch of simple carbohydrates linked together. So a whole bunch of, right, fructose or sucrose or glucose or maltose, a whole bunch of those simple building blocks linked together make this big, huge structure that we call a complex carbohydrate. And complex carbs are things, right, that we think of our breads, our starches, right? Starches, vegetables, all of those contain complex carbohydrates. And these, since they're, right, big, huge molecules, it takes a while to break your body to break them down into their smaller parts to be used for energy. So complex carbohydrates, once again, right, it's a carbohydrate, so our body, the main purpose for it is energy. It gives us more long-term energy. So it makes sense, right? If we are a marathon runner, right, the night before our race, we load up on, right, complex carbs. We eat the pasta, right, and the bread and all that stuff so that we have enough fuel the next day when we're running that marathon, right? It's going to give us lots of stuff, right? But during the race, it would not be beneficial, right, to eat that bowl of pasta in the middle of the marathon. It's not going to, right, be able to be broken down and us be able to use it as energy during that race, right? But during the race, if we needed quick energy, we were starting to get low on energy, we could grab things like that banana, right? Or we could eat that energy bar that's got simple sugars in it, simple carbohydrates, and our body can use those right away during the race to fuel us. All right, next example of an organic substance is a lipid, right? A lipid is a big fancy word for a fat, right? So when we talk about lipids, we're talking about fats. Within these, when we talk about lipids, they once again are an organic compound, so they have carbon and hydrogen bound together. But lipids, for the most part, are hydrophobic, right? They are not soluble in water. The two of them don't mix, right? If we take that cooking oil and dump it into the water, right, it stays, it doesn't dissolve in it. Lipids, right, have lots of different uses within the body. The body needs them, right? You could not live with 0% body fat. You have to have a healthy amount of fat within your body in order to survive. And one thing we do use lipids for, fat for, is stored energy, all right? But that's not one of the main reasons why we take in lipids, right? It's not for stored energy necessarily, but it's very good at making biological membranes, right? Membranes such as the cell membrane. Remember, the cell membrane was made up of a phospholipid bilayer, right? So the membrane of every one of the trillions of cells in your body is actually made up of fat. It makes good waterproof coverings like your skin, right? You pour water onto your skin, it beads and runs right off. It doesn't, right, absorb into it readily like a sponge. So it keeps the water, right, outside, right? If we jump into the pool, so we don't drown, right? All that water doesn't go into us and make us kill us, right? And it also keeps all the water inside of us from escaping out so we don't dehydrate. When we look at it within the body, we typically find it stored in a couple different ways. As fats, right? So it's found in adipose tissue, your fat tissue. Remember, that was connective tissue, right? As oils within your body. So there are oils on your skin, right? Your sebaceous glands secrete oil on your skin. That's what makes the water run off of it, right? You have waxes in your body, right? There's ear wax, right? Cerumen, as it's called, which is wax that comes out of glands within your ears, and that helps protect, right, and keeps things from getting down into your ear. And then we can use these lipid molecules to make steroids, right, and other hormones of your body. So steroid is one type of hormone, and a lot of your hormones within your body are made from fats. So when we look at it, we can break it down into a couple different components or different ways that fat looks. First one is glycerol and fatty acid chains. These make up what we call triglycerides. This is typically how we store fat within our body, right? When we look at a fat cell, it's triglycerides that are stored within there. And what we have here is we have this long chain, oops, it doubled up here, sorry about that, right? We have, right, a phospholipid bilayer here. So we have, right, the phosphate group up here, and the lipid part are these long chains. And these long chains are made up of carbons and hydrogens. Right, here we have a triglyceride. Tri means three, so we have three long lipid chains. Fatty acid chains are what we call them. So here you can see we actually drew out all the carbons and hydrogens. And this would be the glycerol part of that triglyceride. And if we look in here, right, all the bonds between each one of these carbons in the fat part of it are all single bonds. And that's going to be important in a minute when we talk about saturated and non-saturated fats. So, right, how many of you guys have heard about, right, consuming, right, unsaturated fats compared to saturated fats for your diet, right, that they're better for us? Things like, right, eating olive oil compared to, right, butter when we're talking about cooking. So saturated, this picture of this triglyceride is what we call saturated fats. We say it's saturated because every carbon is saturated with another element. I can't add any more things to this carbon. A carbon can only hold 4 different things to it. So here we have, right, 2 hydrogens and 2 carbons attached to this one carbon. Right, it can only hold on to 4 things. It can't hold on to any more. So we say it's saturated. All right. If we now put a double bond in there, okay, oops, we go back, right, to this molecule. So here we can see in this particular fatty acid chain over here, here we had 2 carbons that were not saturated. There's a double bond here. So that means that at this carbon and at this carbon, they're only attached to 3 things instead of 4 things within there. So in this case, this would be an unsaturated fat, which means that we could manipulate, right, this bond could be broken and we could add a 4th thing to this carbon or to this carbon and make it saturated. But if you notice what happened at that bond, it changed its shape. Right, it made it bend like this. That's a unique feature, and that leads into, right, the way that fats, different types of fats between saturated and unsaturated fats look, especially at room temperature. Right, butter is a saturated fat. Right, the fat on your bacon or on your steak before you cook it is a saturated fat. Right? Lard is a saturated fat. All of those are solids at room temperature. Okay, and they're solid because they go back to, right, that other shape, this shape, and there are no bends in it. Everything is nice and parallel and stacks up very densely. It's a very dense molecule, right? There's not a lot of space in between. Whereas an unsaturated fat, right, there's bends in it and there's spaces in between. It's not as dense. So typically unsaturated fats are liquid at room temperature. So things like your olive oil, your canola oil, your corn oil, right, are all unsaturated fats. And then we have these things, you ever heard of trans fats, right? And we talk about trans fats, and typically when we talk about those, we say stay away from them, right? If you are the person that shops for your family and you go to the grocery store and you see, right, and you're looking at the different margarines and the different, right, things that aren't butter, right, all the margarines up there, a lot of them will say, right, does not contain any trans fats, right? And what a trans fat is, all right, within there is now we've chemically altered that fat. Okay, so it was typically wanting to be a liquid. So it was this type of configuration, and we chemically added things to it to break apart this double bond and make it saturated. And by doing that, we alter, right, the natural shape of that fat, which when we first started doing this chemically years and years, over 50 years ago, right, we didn't know that that would cause a problem, right? And the main reason why we did it was out of convenience. It's a lot easier to package and to ship a solid than it is a liquid, so that's why we changed things, right? Like that. But now we're finding out by altering Mother Nature, right? Mother Nature doesn't like to be fooled. So us altering Mother Nature here and changing the chemistry of that fat is it typically causes bad things within our body. So we see things like cancers associated with it and other bad problems and diseases that can happen because of trans fats. So we want to stay away from trans fats. Right. Other type of lipid, right? So we had lipids, triglycerides, right? Phospholipids, where we had these long chains and they could either be saturated or unsaturated. The other way that fats can look are in ring structures like this. And if fats are in a ring structure, this configuration is, that's where we look at cholesterol and steroids. So steroids are the monomers of cholesterol and other hormones within there. Right, and then our third type of macromolecule was proteins. Right, proteins. Right, if we think of that plate of food, right, the meat on there, right, is typically a good source of protein. And when we look at muscle structure and its physiology, you'll understand why within there. But proteins, when we use them, they're really good at making structures. So giving things bulk, right? They're used as regulatory things. Remember those tunnels that I showed you within the cell membrane way back when we were looking at how things move in and out, right? Those were all made out of protein. There are hormones that are made out of protein. So very important. They're a big molecule once again, but they're made up of smaller things called monomers. And the small things that make up protein are amino acids. So they're the building blocks of protein, are my amino acids. And you have 20 different types of amino acids within your body that it can use, right? And we take those 20 different types and put them in all different types of ratios and configurations, and we can make hundreds of thousands of different types of protein within the body. All right, if we look at food sources, okay, food sources within there, right, let me go back, I'm sorry. Things like, right, we said before, your meat, your eggs, right, dairy, seeds, nuts, right, peanut butter is a good source of protein, Other beans, legumes, right, are good sources of protein. It doesn't always have to be meat. You can get protein from plant-based structures as well within there. Typically, one of the problems with a vegan diet, right, a complete vegan diet where you get all your protein 100% from plant materials and nothing from any living or from any animal base, is you don't get all 20 different types of amino acids within there. And you get some other types of, you're lacking typically in some amino acids. So that's where supplementation typically has to come into. So people that are strict vegans typically have to supplement with certain amino acids that just aren't found within plant-based material. Right? When we look at proteins, since they make up structure, right? Structures in your body can take on lots of different forms. So there are four distinct shapes that proteins can come in. First one is a primary structure. So primary, it's also known as linear, right? Is these long chains like this. It can either be as a ribbon, like a string, or it could be sheet-like, like a flat piece of paper. Right? Secondary structure now is it's going to start twisting on itself. So what we call helical. Right? So secondary structure starts to twist. So it can either look like that ribbon that we've now twisted, right? Or that piece of paper that we've folded up now so it looks like a Chinese fan. So it's folded or twisted within there. Right? This picture right here looks very, right, like DNA. DNA is a helical structure. It's because of a protein backbone within it. Right? As I said, it could also look like a Chinese fan where it's pleated, if it's more of a sheet-like structure instead of a thread-like structure. And then we have tertiary. Tertiary structure means it's folded onto itself. So kind of think of, right, we have that ribbon. We can take the Christmas ribbon and we can pull it and have it nice lay flat on the table. And that would be a primary structure. I took that ribbon now and started to twist it, right, and curl it. That would be a secondary structure. Now, if I took that ribbon and tied it up and made a big bow out of it, that's kind of what a tertiary structure is. So it's folding in on itself. And then we have a quaternary structure, which just means we're going to take four of these, right? Quant means four, right? So we're taking four of these and linking them together. So we get this huge structure. So quaternary structure would be like your hemoglobin molecule, right? The molecule that's found in your blood that makes your blood work. So those are our different structures of protein, and they do different things, right? We'll see throughout the year, a lot of them are found on your cell membrane, and it's because of these unique structures, right? They may look very random here, but all of these bends and twists and folds are due to their chemical structure, right? Those bonds. If you remember back in chemistry class, right, how I bond different things together, right? Made it bend to the right or to the left, or twist, or made it bend at 90 degrees as opposed to, right, 45 degrees. So all of that, this is due. It's not random at all. It's very precise within there. All right, of these proteins, we typically say they're fibrous type. So those primary and secondary structures are typically fibrous type proteins. Things like within our connective tissue, we find protein fibers like collagen and elastin. Within the, right, keratin within your nails are all proteins within your hair. All right, and they come on different shapes, right? Some people have really straight hair. Some people have very wavy hair. Some people have very kinky hair, right? And that's all because that keratin proteins within your hair, right? And depending what shape they are, if it's a primary structure, a secondary structure, or a tertiary structure, right, of keratin, and that's based on your DNA, right, will tell you what kind of hair, right, you have. A good example of that. Globular proteins are bigger things, right, that tertiary and quaternary structure, and they make up big things like enzymes and hormones and antibodies, right? So these big, huge structures that we'll look at later on throughout the year that have specific shapes. Right, so here we can look at, here is a enzyme, right? So a functional enzyme here, here's the thing that it's working on. So it needs to be in this shape, right, for these three things to fit into it. If it changes its shape like this and starts to unravel for some reason, now it won't fit into this, right, structure. When we say that a protein has become denatured. So denaturing a protein means we've changed its shape. And things that can change a protein's shape are things like temperature changes, pH changes. Sometimes chemicals can denature proteins within there, right? So that's one reason why your body, right, needs to keep these things, right, within very close parameters, right? It needs to keep your temperature within homeostasis. If we get too far out one way or the other, right, it can cause disease and death. Then one of those reasons is because it starts changing the proteins within our body, right? They start unraveling. Same thing with pH, right? If the pH gets too far out of, remember it was 7.35 to 7.45. If it gets outside of that range, once again, it can start altering our proteins within our body, which make up, right, a huge part of you. Right? Enzymes are globular proteins. So enzymes, right, are protein. That's one reason why we need to have protein within our diet. We can't have a diet with zero protein, or we would not be able to make enough enzymes. Enzymes are what we call biological catalysts. If we remember from chemistry, a catalyst is something that speeds up a chemical reaction. All right. I'm not going to, don't worry about the halo enzyme, right? Don't worry about that second bullet there. I'm not going to hold you to that. But what I do want you to know is that enzymes are catalysts, so they speed up chemical reactions. They speed up the chemical reactions in your body by a lot. We're typically talking at the magnitude of 100,000 to 1,000,000 times faster. All right, it's not that, right, a chemical reaction by adding an enzyme to it is going to be twice as fast. It's going to be 100,000 times faster or 1,000,000 times faster. So they're very important within our body. That's what allows our body to react and make changes so quickly, is because of enzymes. They are extremely specific, right? Remember those proteins, each protein had its own shape to it depending on its chemical structure. So they're very chemical specific. Typically, we're talking about one enzyme for one certain reaction, right? Kind of think of it like the key, right, to your car, right? That key to your car only works on your car. It doesn't work on, right? It's not a skeleton key that works on every car in the parking lot. Right? And when we talk about enzymes, they typically end in ase, right? Remember, sugars, saccharides end in ose, enzymes end in ase. And they typically, the word preceding that is going to be the type of structure it is. So things like ATPase, right? Or dehydrogenase. Those are enzymes, right? A dehydrogenase works on dehydrogen synthesis reactions, right? ATPase works on ATP. And the way that enzymes work is they lower what's called the activation energy. Activation energy is the energy required to make a reaction happen. right? So when we look at this, I'll go back, oops, right? An enzyme is going to bind with what we call the substrate. So that's the chemicals, right? The reactants that it works on if we use our chemistry terms. And a new product is formed, right? And it did that by lowering the activation energy. And then it releases that product. And it can work over again. So here we have, right, the green is this big tertiary looking protein. It's this big structure, right? And here are our reactants. So we want these two things to come together to form this new molecule. What this enzyme does is it helps hold them together closely, right? Picks them up so they are held so close together that now they can bond, right? Form a new bond and form that molecule, and then it releases it, right? As we see here, right, the shape that it started with and the shape that it ended with of the enzyme is exactly the same. So it gets reused again. So this same one, right, picks up two new ones, right, and does this over and over and over again. So enzymes are very efficient in that they're reusable. They're not consumed in the reaction. All right, and they're very efficient in that they speed up reactions on that magnitude of 100,000 to millions of times faster. Right, and then our last bit, right, that we're going to end with today are nucleic acids, where you've heard of these before. That's what DNA and RNA are, right? The Na of both of those is nucleic acid, right? The first part stands for the sugar that's attached to this nucleic acid. So deoxyribose nucleic acid is DNA. Ribosomal nucleic acid is the second one within there. So nucleic acids are found within your DNA and RNA. So that's living things once again. And there are also nucleic acids make up ATP. So that is your right, energy molecule. So nucleic acids are very important, right? Without them, we wouldn't have DNA. Without DNA, right, we could not live. And without energy, we could not live. So nucleic acids are extremely important. Right, here we can see structure, right? So it's a sugar with a nucleotide attached to it. So remember in DNA we had, right, adenine, guanine, cytosine, and tyrosine within there. Within an ATP molecule, right, we have a sugar, and then we have this phosphate on there. So this is that nucleic acid part to it. Over here, the adenosine. So, right. All right, that's the end. I'm going to go ahead and end our slideshow here. All right, and let me go ahead and transition back. So welcome back. All right, long lecture there, but that was looking at chemistry. So we dived into chemistry. That's the extent of chemistry that I want you to know for this class. So hopefully you've had some chemistry courses already and none of that was new to you. But we're not going to dive into, right, a whole semester or year-long course of chemistry. With that, thank you for joining us today, and look forward to talking to you in our next video. So have a great day, and I will talk with you later. Bye now.